All right, on this upcoming test, you will need to read some of the book. Don't expect you to read every chapter, 6 through 10, all the way, and know everything in that. We will focus mainly on chapter 6 and 7. And to help you out with what you need to know in chapter 6 and 7, I have made these videos. So this is the first. This is chapter 6, and we're looking at section 1. And we're just going to go through some common things you need to know about microscopy. So we're going to do a lab on microscopy. Of course, we can't go into the lab and use them, but we can talk about them. You can learn the parts, that sort of thing. So be ready. We'll probably have a test on Monday. And this sort of material will be in there somewhere. Okay, so what we're talking about is microscopy. And there are essentially two types of microscopy: light microscopy, so a light microscope, and an electron microscope. Electron microscope uses electrons. Light microscopes use light or photons. And the electrons are different. We can't see electrons, but we can create a machine that can. And so you can see smaller things with electron, more resolution. You can see than you can with a light microscope. But of course, light microscopes are a lot cheaper. And I'm sure everybody's seen a regular light microscope. The three things you need to know: you'll need to know what the term magnification, resolution, and contrast. If you get a question on the test, you're likely to see these three words used, so you need to know how to use them and know the difference between these words for a test question. Now, size. Here's the light microscope. You can see cells and most bacteria, and even some of the things in the bacteria. But light microscopy is limited usually to bacteria and nothing smaller. We are improving, and we are seeing smaller things, but electron microscopy can see very small things, including like the lipids and the proteins. And that's where you want to use an electron microscope. So here's just another picture. This is in the book, showing you what you can't see. Of course, it's really hard to see things like atoms because you're using electrons in an electron microscope, and electrons are part of atoms. It's really hard to see what's going on when you do that. But the idea here is we can get down to proteins and lipids, which is impressive. You need to know that a light microscope can roughly magnify by about 1,000 times. The standard one that you see is usually only about 450 times, but we've made lots of improvements. And when you get down to those levels, you can begin to see the organelles. So we're going to talk a lot about organelles in this chapter and in the next. So make sure you go back and review organelles. We talked about these in the last chapter. These are the little compartments. All right, there are many types of light microscopes and electron microscopes. So up here are all of your different types of things you can do with a light microscope. You can stain things. You can use filters and different lenses to make it look kind of 3D. You can make it fluoresce shine different types of light on it, that sort of thing. And then the electron microscope, that's the really cool ones where you get to see things in really large detail. These are just some more of the same pictures showing what you can do. Here is unstained, and here's stained, and here are some things so you can see the three-dimensional structures of them. All right, same of that. Now, electron microscopy, these are actually cilia coming out of a cell. These little bumps here are all lipids. So you're wondering what the membrane looks like. This is it. And then these are things coming out of the membrane of the cell. Like I said, electron microscopes, there are two types: SEMS, scanning electron microscope, and transmission electron microscopes. This one tries to make a 3D image, very similar to what we just saw. The transmission tries to look inside of the organism. So if you were studying the organelles of an organism while it's still alive, you would use a transmission electron microscope to see what the inside is doing and actually watch it move and change shape and things like that. You would use a transmission electron microscope. So you might get some questions about why would I want to use a transmission electron microscope or over a scanning electron? Like, well, we want to find out what's going on inside the cell. Here we're looking at more or less what's going on at the surface of the cell. So these are the two. You might check those out. All right, and light microscopy has improved recently, so we need to recognize that light microscopes are getting as good as the worst electron microscopes. So there's a resurgence of light microscopy because the technology with digital cameras and things like that have improved so much. You can now see as well as you can with the worst electron microscopes, and these are a lot cheaper, of course. All right, and that will do it for this little section. Just keep in mind our test is probably going to be on Monday.